how do you envision how this might work in humans? To what extent, if it does, the transfer of RNA? We know that it does a little bit. To what effect we don't know, but how do you envision where this might play out in, say, the next 10 years?
Mhm. So it's very hard to study in humans. There
Yes.
are people who say it can't happen in humans. Sometimes use um, this sort of a logic. They say, no, in worms it makes sense. As I said, it's not controversial anymore with coelacanths. They say in coelacanths it makes sense because the generation time is so short, it's just 3 days. So it makes sense to prepare the next generations for what the parents experienced.
Hmm.
The likelihood that the environment that the parents live in would be similar to the environment of the progeny is high. So it makes sense to prepare them for
Right.
what you expect. Okay,
Hmm.
and also they don't move so far because they're small. So
Hmm.
maybe environment say my kids, might live in a different country. Who knows? It will take 20 years. Uh, but I think that this is not necessarily, a great um, explanation or a reason because there are different scales to things. Like if you're talking about viruses, we encounter the same viruses for many generations or similar
Right.
families of viruses. So it doesn't have to be a real reason for it not to occur. It's very hard to study in humans because we can do it's hard to do experiments. We don't want to do experiments on humans,
Hmm.
and the generation time is so long, and we are very different genetically. So the worms that we study in the lab are almost identical in terms of their DNA, and also they live in a very defined condition. So it's easy to separate nature from nurture. In humans, that's hard to do. Another reason why people say it can't happen in humans is that in C. elegans, it really depends on these RNA-dependent RNA polymerases that amplify the small RNAs,
Yes.
and we humans don't have them.
Right.
However, there are other mechanisms that humans have that C. elegans don't have, like DNA methylation.
Right.
And also, I just, gave you a scoop that it does happen in RNA in planaria, and planaria don't have RNA-dependent RNA polymerases as well. Okay? So there could be different tricks. There are all kinds of feed-forward mechanisms that might maintain gene regulatory responses. So I really can't say anything about humans aside from really speculations. I think that we will get there someday to understand. And probably the key is establishing a very good system because even the experimental design of such an experiment is not clear.